Bismillahirrahmanirrahim. I am Dr. Zahra Noreen, course instructor of Plant Physiology 1, BOTN 3119. Today we will talk about our second last topic of photosynthesis, carbon dioxide concentrated mechanisms. Many plants either do not photorespire at all or they do so to only a limited extent. These plants have normal rubiscos and their lack of photorespiration is a consequence of mechanisms that concentrate carbon dioxide in the rubiscose environment and thereby suppress the oxygenation reaction. Three mechanisms for concentrating carbon dioxide at the site of carboxylation. Number one is carbon dioxide pumps at the plasma membrane. Number two is C4 photosynthetic carbon fixation or C4 pathway. And number three is carboxylation acid metabolism, also known as CAM pathway. The last two of these carbon dioxide concentrating mechanisms, C4 and CAM, are found in some angiosperms and involve add-ons to the Calvin cycle. Plants with C4 metabolism are often found in hot environments and CAM plants are typical of desert environments. We will examine each of these two systems after we consider the mechanism of carbon dioxide pump found in aquatic plants that has been studied extensively in unicellular cyanobacteria and algae. First of all, carbon dioxide concentrating mechanism 1, that is algal and cyanobacterial pumps. When algal and cyanobacterial cells are grown in air enriched with 5% carbon dioxide and then transferred to a low carbon dioxide medium, they display symptoms typical of photorespiration, that is oxygen inhibition of photosynthesis at low concentration of carbon dioxide. But if the cells are grown in air containing 0.03% of carbon dioxide, they rapidly develop the ability to concentrate inorganic carbon dioxide plus bicarbonate internally. Under these low carbon dioxide conditions, the cells no longer photorespire because they do concentrate the amount of carbon dioxide by a mechanism they have. At the concentrations of carbon dioxide found in aquatic environments, rubisco operates far below its maximal specific activity. Marine and freshwater organisms overcome this drawback by accumulating inorganic carbon by the use of carbon dioxide and bicarbonate pumps at the plasma membrane. ATP derived from the light reactions provide the energy necessary for the active uptake of carbon dioxide and bicarbonate. Total inorganic carbon inside some cyanobacterial cells can reach concentration of up to 50 millimolar. Recent work indicates that a single gene encoding a transcription factor can regulate the expression of genes that encode the components of carbon dioxide concentrating mechanism in algae. The protein that function as carbon dioxide bicarbonate pumps are not present in cells grown in high concentration of carbon dioxide, but are induced upon exposure to low carbon dioxide concentration. The accumulated bicarbonate is converted to carbon dioxide by the enzyme carbonic anhydrase, and the carbon dioxide enter the Calvin cycle. The metabolic consequences of this carbon dioxide enrichment is suppression of the oxygenation of the ribulose 1,5-bisphosphate and hence also suppression of the photorespiration. The energetic cost of this adaptation is the additional ATP needed for concentrating the carbon dioxide. Carbon dioxide concentrating mechanism 2, the C4 carbon cycle. There are differences in the leaf anatomy between the plants that have a C4 carbon cycle called C4 plants and those that photosynthesize solely via the Calvin cycle or the Calvin photosynthetic cycle, that is C3 plants. A cross section of a typical C3 leaf will leave one major cell type that has chloroplast, and that is the mesophyll tissue. In contrast, a typical C4 leaf has two distinct chloroplast containing cell types. One is the mesophyll cells and the other one is the bundle sheath cell. Or the presence of the chloroplast in the bundle sheath cells, and also this type of the anatomy which the C4 plants have is known as Kranz anatomy. Kranz is a German word which means wreath. There is considerable anatomical variations in the arrangement of the bundle sheath cells with respect to mesophyll and vascular tissues. In all cases, however, the operation of the C4 cycle requires the cooperation effort of both the cell types, the mesophyll and the bundle sheath. No mesophyll cell of a C4 plant is more than 2 or 3 cells away from the nearest bundle sheath cell. In addition, an extensive network of plasmodesmata connect mesophyll and bundle sheath cells, thus providing a pathway for the flow of metabolites between the cell types. So, इनके दरमियान में कोई भी मुश्किल पेश नहीं आती अगर इन दो सेल्स को आपस में कॉपरेशन की ज़रूरत है फॉर द ऑपरेशन ऑफ C4 साइकिल द mesophyll cells and the bundle sheath cells. तो प्लाज्मोडेस्माटा के न्यूमरस कनेक्शन होने की वजह से इन दोनों के दरमियान में मेटाबोलाइट की जो एक्सचेंज है जो फ्लो है वो जारी रहता है. Here is a cross sections of leaves showing the anatomic differences between C3 and C4 plants. Diagram A representing a C4 monocot, Saccharum officinarum, sugarcane, in which you can see there is no differentiation of the mesophyll tissue into palisade layer and spongy parenchyma. And also notice the large size bundle sheath cells around the vascular bundles. In case of diagram B, it's a C3 monocot, poa species, a grass. You can notice here that it can be differentiated into palisade layer and spongy parenchyma. These are the elongated palisade layers and these cells are the spongy parenchyma. And also see the difference of the size of bundle sheath cells as compared to the C4 anatomy. In case of diagram C, it's C4 dicot, Flavaria australasica, Austraceae family, a member of the Austraceae family. Again, you can see there is no differentiation of the mesophyll tissue into palisade layer and spongy parenchyma, and also the bundle sheath cells are large in size. One thing is more noticeable that either the case of C4 dicot or the case of C4 monocot, the mesophyll tissue is not away from the nearest bundle sheath cells, 1 to 2 cells, only away 1 to 2 cells from the nearest bundle sheath cells. And this is not in case of C3 plant. In case of D diagram, it's a schematic diagram representing 3-dimensional model of a C4 leaf, showing the presence of chloroplast in the bundle sheath cells, which is not present in case of C3 plants, and also the absence of differentiation of the mesophyll tissue into palisade layer and spongy parenchyma. So these two features makes C4 plant with different anatomy as compared to a C3 plant. Here in diagram E, you can see the presence of numerous plasmodesmata connection which are required to connect the two types of the cells. In case of C4, the mesophyll cells and the bundle sheath cells. So a strong connection is required because the metabolites are moved from one cell to another, and this movement of the metabolites also required for the operation of the C4 cycle. Malate and aspartate are the carboxylation products of the C4 cycle. जब मैंने आपको C2 and C3 pathway पढ़ाया था, मैंने उस टाइम भी आपको बताया था कि why we are saying those pathway as C2 and C3 pathway, because the first stable metabolites in those cycles were C2 2 carbon containing, or C3, a 3 carbon containing compound. Similarly, here is the case in case of C4 cycle. um, the first intermediary compound, which is the stable intermediary compound, is a 4 carbon containing compound. It can be malate, and it is also the aspartate. So early labeling of the C4 acids was first observed in carbon dioxide isotope labeling studies of sugarcane by H.P. Kortschaks and colleagues, and of the maize by Karpilov and co-workers. शुगर केन को कोरशैक्स और उसके कलीग्स ने स्टडी किया, जबकि मेज़ को कापिलोव और उसके कोलीग्स ने, को वर्कर्स ने जो है वो स्टडी किया विद द हेल्प ऑफ आइसोटोप ऑफ कार्बन डाइऑक्साइड. एंड व्हेन leaves were exposed for a few seconds to isotopic carbon dioxide in the light, 70 to 80% of the label was found in C4 acids, malate and aspartate. A pattern which is very different from one observed in the leaves that photosynthesize solely via the Calvin cycle. उनमें 70 to 80% जो है वो malate and aspartate produce नहीं होता first few seconds में. In pursuing these initial observations, Hatch and Slack elucidated that what is now known as C4 photosynthetic carbon cycle, or the C4 cycle. They established that the C4 acids, malate and aspartate, are the first stable detectable intermediates of the photosynthesis in leaves of sugarcane, and that carbon atom 4 of the malate subsequently becomes the carbon atom 1 of the 3-phosphoglycerate of Calvin cycle. The primary carboxylation in these leaves is catalyzed not only by rubisco, but another enzyme which is known as PEP carboxylase, or phosphoenol pyruvate carboxylase. The manner in which the carbon is transferred from carbon atom 4 of malate to carbon atom 1 of 3-phosphoglycerate become clear when the involvement of the mesophyll and bundle sheath cells were elucidated. The participating enzymes occur in one of the two cell types. The PEP carboxylase, which firstly incorporate the carbon dioxide. Actually, this enzyme and the pyruvate orthophosphate dikinase, both these enzymes are restricted to mesophyll cells, and the decarboxylase enzyme, which release the carbon dioxide, and the enzymes of the complete Calvin cycles are confined to the bundle sheath cells. So with this knowledge, Hatch and Slack were able to formulate the basic model of the cell, which I will explain in the coming slide. Here is the basic C4 photosynthetic carbon cycle. Involves the 4 stages in 2 different cell types, mesophyll cell and the bundle sheath cells. These two passages showing the plasmodesmatal connection between these two cells. In step 1, the atmospheric carbon dioxide is fixed into mesophyll cells into a 4 carbon containing compound. In step 2, this 4 carbon containing compound get transported from the mesophyll cell to the bundle sheath cells, and in the bundle sheath cells, this 4 carbon containing compound get decarboxylated and release the carbon dioxide, which is utilized to operate the Calvin cycle in the bundle sheath cell. So Calvin cycle does not operate in mesophyll cells, it operates in the bundle sheath cell. And then afterward, the C4, after releasing this carbon dioxide, will be converted into a C3 acid, and this C3 acid will move back to the mesophyll cell, and here it will regenerate again the precursor phosphoenol pyruvate, which is ready to receive another molecule of carbon dioxide and incorporate into a C4 acid. As I always give you a summary of the reactions along with their enzymes, so here is the list of the enzymes involved in C4 photosynthetic carbon cycle along with their reactions. The C4 cycle concentrate carbon dioxide actually in the bundle sheath cells. So the basic C4 cycle consists of 4 stages, as I told you in the previous slide. Number 1 is the fixation of the carbon dioxide into a C4 acid, the malate, or it can be aspartate. and then the transportation of the C4 to the bundle sheath cells. And in step 3, the C4 will decarboxylate and release carbon dioxide, which will be utilized for the Calvin cycle. And in step 4, the transport of the C3 acid, it can be pyruvate or alanine, and which will be transported back to the mesophyll cell and regenerate the carbon dioxide acceptor phosphoenol pyruvate. One interesting feature of the cycle is that regeneration of the primary acceptor phosphoenol pyruvate, which consumes two high energy phosphate bonds, one in the reaction catalyzed by the pyruvate orthophosphate dikinase and another in the conversion of ATP and AMP to ADP catalyzed by the adenylate kinase. So two extra ATP are required to operate C4 cycle along with C3 pathway in C4 plants. So shuttling of the metabolites between the mesophyll and bundle sheath cells is driven by diffusion gradients along numerous plasmodesmata connection, and transport within the cells is regulated by concentration gradient and the operation of the specialized translocator at the chloroplast envelope. The cycle thus efficiently or effectively shuttle carbon dioxide from the atmosphere into the bundle sheath cells where the Calvin cycle operates, and this transport process generates a much higher concentration of carbon dioxide in the bundle sheath cells than would occur in equilibrium with the external atmosphere. That is why this elevated concentration of carbon dioxide at the carboxylation site of the Rubisco results in the suppression of the oxygenation of ribulose 1,5-bisphosphate and hence of the photorespiration. क्योंकि photorespiration को होने के लिए मैंने आपको कंडीशंस बताई थी कि ऑक्सीजन की कंसंट्रेशन अगर हाई हो और कार्बन डाइऑक्साइड की कंसंट्रेशन अगर लो हो तो वो फोटो रेस्पिरेशन की तरफ रुबिस्को को जो है वो इन्वॉल्व कर देती है. बट क्योंकि इन केस ऑफ C4 साइकिल वो कभी भी कार्बन डाइऑक्साइड की कंसंट्रेशन को लो नहीं होने देते, जहाँ पे एक्चुअली कार्बन डाइऑक्साइड ने फिक्स होना है कैल्विन साइकिल में, उस साइट में बंडल शीट सेल्स में, क्योंकि ये कार्बन डाइऑक्साइड की कंसंट्रेशन को लो नहीं होने देते ये साइकिल, दैट इज़ वाई C4 प्लांट्स में फोटो रेस्पिरेशन भी नहीं होती. So this discovered in the tropical grasses, sugarcane and maize, and the cycle is now known to occur in 16 families of both monocotyledons and dicotyledons, and it is particularly prominent in gramineae, corn, millet, sorghum, sugarcane, Chenopodiaceae like in Atriplex, and Cyperaceae family जैसे कि sedges हैं उनमें ये commonly आपको नज़र आता है. ओवरऑल अगर हम देखें तो 1% ऑफ ऑल नॉन स्पीशीज़ जो प्लांट की पाई जाती हैं उनमें से 1% जो है उनमें C4 मेटाबॉलिज्म जो है वो ऑपरेट होता है. There are 3 different variations of the basic C4 pathway that occurs in different species, and these variations differ principally in C4 acid. जैसा कि मैंने आपको बताया कि 4 कार्बन पे मुश्तमिल जो है वो एसिड बनना है, तो वो एसिड मैलेट भी हो सकता है और एस्पार्टेट भी हो सकता है. तो इसी की बुनियाद पर और यूज़ होने वाले एंजाइम्स जो कि डिकार्बोक्सिलेशन में इस्तेमाल होते हैं इन बंडल शीट सेल्स, इन दोनों की बुनियाद पे C4 पाथवे जो हैं ये इनमें 3 तरह की वेरिएशंस जो हैं वो पाई जाती हैं, जो कि मैं आगे एक्सप्लेन करूँगी. फर्स्ट ऑफ ऑल हम जो इसका बेसिक मैकेनिज्म है उसको देख लेते हैं, एंड देन फिर हम उसकी वेरिएशंस को देख लेते हैं. So here is the C4 photosynthetic, a typical C4 photosynthetic pathway in which the hydrolysis of 2 ATP drives the cycle in the direction of the arrows you can see here, and thus pumping the carbon dioxide from the atmosphere to the Calvin cycle of the chloroplast from the bundle sheath cells. The cycle starts with the incorporation of carbon dioxide in the presence of enzyme carbonic anhydrase into bicarbonate, and this bicarbonate is utilized by PEP, phosphoenol pyruvate, in the presence of enzyme PEP carboxylase, which will convert this 3 carbon containing compound with the help of this bicarbonate into oxaloacetic acid. Oxaloacetic acid then, in the presence of enzyme malate dehydrogenase, you utilize 1 NADPH and will convert into malate, 4 carbon containing compound again, and this malate is then transported or translocated from the mesophyll cells to the bundle sheath cells, and here it will release the carbon dioxide with the help of malic enzyme, and this malic enzyme will convert the malate into pyruvate, a 3 carbon containing compound, because the malate was a 4 carbon containing compound, and after releasing 1 carbon dioxide it will be converted into 3 carbon containing compound. This released carbon dioxide will be utilized in the Calvin cycle. So you can see that the Calvin cycle in case of C4 plants operates in the bundle sheath cells, while in case of C3 plants it always operates in the mesophyll cells. The 3 carbon containing pyruvate then translocated back to the mesophyll cells, where it converted into PEP with the help of enzyme pyruvate phosphate dikinase, which will utilize of course an ATP molecule and convert it into AP and PPI pyrophosphate, and pyrophosphate will be further catalyzed by phosphatase enzyme into inorganic phosphate. This AMP with the help of another ATP in the presence of enzyme adenylate kinase will be converted into 2 ADP. So technically 2 ATPs are required. These 2 ATPs are required for the operation of C4 pathway, which are extra needed. Otherwise, 1 NADPH is also required, but this NADPH is compensated with the production of this NADPH in the mesophyll cells, bundle sheath cells. So the PEP, which is formed from the pyruvate, is now ready again to react with the bicarbonate and form another molecule of oxaloacetate. Here is the energetics of the C4 photosynthetic cycle. You can see here that overall, if we see here two steps which involve the ATP. One is the pyruvate, which utilize the ATP and convert the pyruvate to phosphoenol pyruvate, and AMP and PPI are generated as well, and this happened in the mesophyll cells. And PPI, I told you, with the help of enzyme phosphatase um, will be catalyzed into 2 inorganic phosphate molecule, and AMP, which was generated in this reaction with the help of another ATP, will be converted into 2 ADP. So overall we can say that 2 ATP are required for the C4 pathway to operate. The concentration of carbon dioxide in the bundle sheath cells has an energy cost. The net effect of the C4 cycle is to convert a dilute solution of carbon dioxide in the mesophyll cells into a concentrated carbon dioxide solution in the cells of the bundle sheath. So studies with the PEP carboxylase deficient mutant of Amaranthus edulis clearly showed that the lack of an effective mechanism for concentrating carbon dioxide in the bundle sheath markedly enhances photorespiration in the C4 plant. So क्योंकि amaranthus जिस मैंने आपको बताया इट्स अ डेफिशिएंट म्यूटेंट इसका जो लिया गया, जिस डेफिशिएंट म्यूटेंट किस चीज़ का है कि इसमें पेप कार्बोक्सिलेज एंजाइम जो है वो नहीं पाया जाता, क्योंकि पेप कार्बोक्सिलेज एंजाइम ही यूटिलाइज हो रहा है फॉर कंसनट्रेटिंग कार्बन डाइऑक्साइड. दैट इज़ वाई these mutants were unable to get rid of from photorespiration. So उनके अंदर enhanced level जो है photorespiration के वो देखे गए. हालांकि ये Amaranthus edulis जो है वो normal जो plant है wild type जो है वो C4 plant है. So thermodynamics tells us that work must be done to establish and maintain the carbon dioxide concentration gradient in the abundant sheath. And this principle also applies to the operation of the C4 cycle. From a summation of the reaction involved, we can calculate the energy cost to the plant, and the calculation shows that carbon dioxide concentrating process consumes 2 ATP equivalents to 2 high energy bonds per carbon dioxide molecule transported. Thus, the total energy requirement for fixing carbon dioxide by the combined C4 and C3 Calvin cycle is 5 ATP, because 3 ATP were required for Calvin cycle, and 2 NADPH which were required for Calvin cycle, and now 2 additional ATP are required to operate C4 cycle. So collectively 5 ATP and 2 NADPH required in C4 plants to fix one molecule of carbon dioxide. Because of this high energy demand, C4 plants photosynthesizing under non-photorespiratory condition, that is high carbon dioxide concentration and low oxygen level, require more quanta of light per carbon dioxide than C3 leaves do. In normal air, the quantum requirement of the C3 plants changes with factors that affect the balance between the photosynthesis and photorespirations, such as temperature. तो टेम्परेचर की फ्लक्चुएशन के हिसाब से C3 की जो डिमांड है एनर्जी की, वो भी चेंज होती रहती है. अगर C3 अकेला पाथवे चलेगा तो उसकी एनर्जी की डिमांड ज़्यादा नहीं होगी, बट अगर टेम्परेचर हाई हो जाएगा तो फोटो रेस्पिरेशन भी ऑपरेट होगा. और मैंने आपको बताया था कि फोटो रेस्पिरेशन में एक्स्ट्रा ATP जो है वो रिक्वायर्ड होते हैं. तो इन दैट केस फिर सेम प्लांट है लेकिन हाई टेम्परेचर पे फोटो रेस्पिरेशन होने की वजह से उसको ज़्यादा ATP जो है वो यूटिलाइज करने पड़ते हैं. और लेकिन अगर उसके बरक्स हम C4 प्लांट्स को देखें तो क्योंकि इनमें फोटो रेस्पिरेशन होती ही नहीं है, उसको अवॉइड कर सकता है ये, और बिल्ट इन है कि ये अवॉइड कर सकता है फोटो रेस्पिरेशन को. दैट इज़ वाई इसकी जो क्वांटम रिक्वायरमेंट, ऑल दो एक नॉर्मल C3 प्लांट से इसकी क्वांटम रिक्वायरमेंट जो है वो हाई है, लेकिन वो कांस्टेंट रहती हैं, वो फ्लक्चुएट नहीं करती हैं विद रिस्पेक्ट टू एनवायरमेंटल कंडीशंस. Light regulates the activity of the key C4 enzymes. Light is essential for the cooperation of the C4 cycle because it regulates several specific enzymes. जैसा कि हमने C3 पाथवे में भी देखा, C2 पाथवे में भी देखा कि लाइट का एक बहुत इम्पोर्टेंट रोल है एंजाइम्स की एक्टिवेशन एंड डीएक्टिवेशन के हवाले से. फॉर एग्जांपल जो एक्टिविटीज़ हैं पेप कार्बोक्सिलेज की और NADP मैलेट डिहाइड्रोजिनेज की एंड पायरुवेट ऑर्थोफॉस्फेट डाइकाइनेज की, जो कि C4 के एंजाइम्स हैं, दीज आर रेगुलेटेड इन रिस्पॉन्स टू वेरिएशंस इन द फोटोन फ्लक्स डेंसिटी बाय टू डिफरेंट प्रोसेसेस. रिडक्शन ऑक्सीडेशन ऑफ थायल ग्रुप्स के ज़रिए भी ये एक्टिवेट डीएक्टिवेट होते हैं, और फास्फोराइलेशन डीफास्फोराइलेशन के मैकेनिज्म के ज़रिए भी ये एक्टिवेट डीएक्टिवेट हो सकते हैं. अब हम वन बाय वन इन एंजाइम्स को देखते हैं कि ये किस तरीके से होते हैं. अगर हम बात करते हैं NADP मैलेट डिहाइड्रोजिनेज एंजाइम की, सो दिस इज़ रेगुलेटेड बाय द थायरोडॉक्सिन सिस्टम ऑफ द क्लोरोप्लास्ट, जो कि मैं आपको ऑलरेडी C3 पाथवे में की रेगुलेशन में एक्सप्लेन कर चुकी हूँ. और द एंजाइम इज़ रिड्यूस्ड फॉर्म में जो एक्टिवेटेड होता है अपॉन एल्यूमिनेशन ऑफ लीव्स, यानी कि लाइट के प्रेजेंस में एक्टिवेट होगा, और ऑक्सीडाइज्ड हो जाएगा यानी कि इनएक्टिवेट हो जाएगा अपॉन डार्कनिंग. The second enzyme is PEP carboxylase, which is activated by the light-dependent phosphorylation and dephosphorylation mechanism. लेकिन कब ये एक्टिवेट होता है और कब ये इनएक्टिवेट होता है, मतलब either phosphorylation is required for activation or dephosphorylation is required for activation, ये सब के लिए अभी जनरलाइज नहीं हो सका. इसके लिए हमारे पास sufficient knowledge अभी मौजूद नहीं है. The third regulatory member of the C4 pathway, pyruvate orthophosphate dikinase, this enzyme is rapidly inactivated by an unusual ADP dependent. Normally हम देखते हैं कि enzymes जो हैं वो ATP dependent होते हैं, but this enzyme is ADP dependent phosphorylation जो है उसके ज़रिए ये जो है वो inactivate होता है. और जब इसको फोटोन फ्लक्स डेंसिटी जो है वो प्रॉपर मिलती है, यानी कि लाइट की प्रेजेंस में ये जो है वो एक्टिवेट हो जाएगा, और अगर फोटोन फ्लक्स डेंसिटी जो है वो ड्रॉप हो जाती है ये इन द प्रेजेंस ऑफ एब्सेंस ऑफ लाइट यानी कि डार्कनेस में, तो ये इनएक्टिवेट हो जाता है. एक्टिवेशन इज अकम्प्लिश्ड बाय फास्फोराइलेटिक क्लीवेज ऑफ दिस फॉस्फेट ग्रुप. मीन्स के जब उसने एक्टिवेट होना होता है तो डीफॉस्फोराइलेशन हो जाती है, और जब डीएक्टिवेट या इनएक्टिवेट होना होता है तो एक्चुअली वो फास्फोराइलेट हो जाता है. So both reactions, phosphorylation and dephosphorylation, appears to be catalyzed by a single regulatory protein. In hot, dry climates, the C4 cycle reduces photorespiration and water loss. तो नॉर्मली अगर हम C4 प्लांट्स को देखें तो इनकी जो आप कह लें कि जो फेवरेबल कंडीशंस हैं जिनके अंदर ये ग्रो करते हैं, तो हम जिन एनवायरनमेंट्स के ये प्लांट्स हैं तो अगर हम उनको देखें तो वो नॉर्मली हॉट एंड ड्राई क्लाइमेट्स के ये प्लांट हैं। और टू फीचर्स जो हैं C4 साइकिल की इन C4 प्लांट्स overcome the deleterious effects of the high temperature on photosynthesis that were noted earlier. First is the affinity of the PEP carboxylase for its substrate bicarbonate is sufficiently high that the enzyme is saturated by bicarbonate in equilibrium with their levels of the कार्बन डाइऑक्साइड। मतलब बहुत कम कार्बन डाइऑक्साइड, जैसे कि हम जानते हैं कि एयर में भी ऑक्सीजन की निस्बत कार्बन डाइऑक्साइड की जो कंसनट्रेशन है वो कम ही होती है, लेकिन दैट इन्वायरमेंट इज सफिशिएंट टू कंसनट्रेट द कार्बन डाइऑक्साइड इन केस ऑफ C4 प्लांट्स। फर्दरमोर द सबस्ट्रेट जो है बाइकार्बोनेट, इसका ऑक्सीजन के साथ कोई कंपटीशन भी नहीं है। एज इन केस ऑफ C3 प्लांट उसमें ऑक्सीजन और कार्बन डाइऑक्साइड का आपस में कंपटीशन होता है फॉर रुबिस्को एंजाइम, लेकिन यहाँ पे क्योंकि सबस्ट्रेट जो है वो चेंज हो चुका है, कार्बन डाइऑक्साइड नहीं है बल्कि बाइकार्बोनेट है, तो उसका ऑक्सीजन के साथ कोई कंपटीशन भी नहीं है। So this high activity of PEP carboxylase enables the C4 plants to reduce the stomatal aperture and thereby conserve the water while fixing the carbon dioxide at rates equal to or greater than those of the C3 plants. तो even hot environment में हो तो ये किस तरीके से पानी की बचत कर सकते हैं कि ये कार्बन डाइऑक्साइड को, क्योंकि ये ऑलरेडी कन्वर्ट कर चुके होते हैं बाइकार्बोनेट में, तो स्टोमेटल अपर्चर को ये कदरे बंद कर सकते हैं एज कम्पेयर टू C3 प्लांट्स। दैट इज़ वाई, क्योंकि स्टोमेटल अपर्चर अगर पूरा ओपन नहीं होगा तो ट्रांसपरेशन ज़्यादा नहीं होगी, और जब ट्रांसपरेशन ज़्यादा नहीं होगी तो वाटर लॉस भी नहीं होगा। सेकेंड बेनिफिशियल फीचर जो है जो C4 प्लांट के हैं वो सुप्रेशन ऑफ फोटो रेस्पिरेशन है। और वो कैसे होती है वो ऑलरेडी मैं आपको डिस्कस कर चुकी हूँ कि बंडल शीथ सेल्स में कार्बन डाइऑक्साइड जो है वो कंसनट्रेटेड हालत में ऑल टाइम मौजूद रहती है और उसकी सप्लाई जो है वो कॉन्टीन्यूस रहती है, और फोटो रेस्पिरेशन तो तभी होगी ना जब कार्बन डाइऑक्साइड के लेवल जो है वो डिक्रीज होंगे। तो क्योंकि C4 प्लांट्स में कार्बन डाइऑक्साइड के बंडल शीथ सेल्स में जहाँ पे कैल्विन साइकिल ऑपरेट हो रहा होता है वहाँ पे इसके लेवल्स क्योंकि डिक्रीज नहीं होते, दैट इज़ वाई फोटो रेस्पिरेशन भी नहीं होती। So these features enable the C4 plants to photosynthesize more efficiently at high temperature than C3 plants, and they are probably the reason for the relative abundance of the C4 plants in the drier and hotter climates. Depending on their natural environment, some plants show properties intermediate between strictly C3 and C4 plants. मतलब आप उनको ये नहीं कह सकते कि वो strictly C3 हैं या वो स्ट्रिक्टली C4 हैं, बल्कि वो एक ऐसे प्लांट होते हैं जो कि जिनके दरमियान में ये एबिलिटी होती है कि वो C3 की तरह भी बिहेव कर सकते हैं और C4 की तरह भी बिहेव कर सकते हैं। This is all about today's lecture. If you have any question or query, you can ask on GCR, WhatsApp, or you can send me email. These are the suggested readings. As always, I give you the name of the books from where the material collected for the preparation of the lecture. Thank you very much.